Okay, guys. Today we're going to talk about viruses, and our big picture for today is we really want to talk about how viruses are so much different than prokaryotic and eukaryotic cells, and how different they are in regards to um, that they are non living and why they are considered to be non living. Um, uh, I don't want to call them organisms because they're not living, but non-living entities that are also pathogens. Okay, so that's our big picture today. We really want to compare how different they are from prokaryotic and eukaryotic cells, and mostly in the realm of why are viruses non-living, and their life cycles or replication cycles, since they're non-living, and what that means, and all the different viruses. And it's quite interesting. I really enjoy viruses. It's one of my favorite topics. So let's go. Okay, so viruses. Viruses are not cells. Okay, so this is a big picture item. Okay, and they're not living. And here is why. So remember when we talked about, like, in the first week of or second week of school, we talked about the characteristics of living things, and we talked about how viruses are non-living. Well, they're non-living because they have a lot of structural differences between a cell and a virus. So even a prokaryotic cell, which is quite smaller than a eukaryotic cell, which you guys should know, viruses aren't a cell. They are just a protein capsid with some kind of genetic information in there, right? So their structural differences include there's no nucleus, there's no cell membrane, and there are no organelles in a virus. Okay. There are also functional differences. This is where we talk about more like characteristics of life. They cannot grow, however, they can adapt, but they cannot grow on their own. They do not respire, meaning they don't have any kind of cellular respiration to get their own energy, right? They're going to utilize their host's energy. They do not develop on their own. Again, they can evolve, but and they can adapt, but they're going to use your host cell's genetic information in order to do that. Okay. And we're going to talk a little bit about that when we talk about lytic versus lysogenic cycles. And they do not reproduce. We say they replicate. Okay. So they do not grow. They do not have like any kind of cellular respiration or metabolism. They do not develop on their own, and they do not reproduce. We say they replicate because remember, they must have a host cell in order to make more viruses. Okay. So that is a big picture item. We must understand that, if anything, today. Okay, so viral structure, like I said earlier, remember, a virus is not a cell. Okay, it is just a capsid, which is a protein coat. Okay, and that capsid includes, um, and sometimes they have an envelope on the outside, like this one does, but a lot of times that capsid includes these proteins on the outside too that actually help bind the virus to the cell, which is, you probably heard a lot about that during COVID-19. The new vaccine. Does the vaccine actually help against the new evolving strains of COVID? Because that vaccine was targeting these outer proteins on the cell, or sorry, on the virus that connect to the cell. Right. So you probably heard a little bit about that. It has some kind of genetic information that is also important. So please write that down. I know that's not underlined, but please write that down. It does have some kind of nucleic acid or genetic information. It can be DNA or RNA, another difference from eukaryotic and prokaryotic cells, whereas all of our genetic information, our blueprint is always going to be DNA, okay, for cells and living organisms. Some viruses have an additional protective coating called an envelope. These are mostly a category of viruses that we call influenza viruses or flu viruses. They all have envelopes. It makes them a little bit more protected. However, it doesn't allow them to stay on surfaces for very long because the surface tension of water will actually pull that apart. Little fun fact for you. Okay, so replication versus reproduction. Something we were kind of talking about before. Remember, viruses do not reproduce as they need a host to actually make more. So we say they replicate only. Okay, they require a host in order for that to happen. Now, you can do that in a bunch of different ways, right? Like you can actually embed your viral genetic information into the host DNA and lay low for a little while and then actually cause an outbreak or an infection, or and that's called a lysogenic virus. I'm going to talk about that in a second. Or you can do it like more immediate. So the virus infects the cell just knows, hey, I need to replicate all of this genetic information, makes a whole bunch of tiny viruses, and then actually causes an infection. So we'll talk about that in a second. The host is a living cell that provides all the material the virus needs to replicate. So remember, the host has to be a living organism in order for that to happen. Okay, so that doesn't necessarily mean just animals. There are viruses that infect plants, or viruses that infect bacteria, and we call them bacteriophages, and we'll talk about that. And of course, there are viruses that infect animals only, and we actually classify viruses by that as well. Another way we classify viruses is shape. Okay, so you do need to know the shape of the virus and an example, and I have those underlined. That's all you really need to know. So the shape allows viruses to attach to certain types of cells. Like I said, remember I talked about those outer proteins outside of the capsid that helps it allows that virus to attach to that cell. So you're going to have a specific shape to attach to certain types of cells. So here we have a filovirus, which doesn't really have any distinct shape, and it's a really weird one. Our example is the Ebola virus right here on the right. It kind of looks like a winding snake type thing. Remember, viruses are much smaller, okay, than even that of a prokaryotic cell. So this is quite magnified with most likely a TEM or an SEM type microscope. So Ebola is our example for a filovirus. Ebola causes massive blood loss through pretty much all orifices. It's pretty rampant in certain parts of Africa, and can be spread through sweat or any kind of bodily fluid. So it's extremely contagious. I wouldn't say it's not airborne or anything like that, but it can be extremely contagious. There was even a couple of cases of Ebola, I believe, in Dallas at one time. And yeah, so it can be very contagious. A polyhedral virus is kind of looks like a hexagon, and it's gonna has those little proteins on the outside right here. The herpes virus is a example of that one. The herpes virus you have two different types of herpes virus. You have a herpes virus, simplex virus 1, HSV1. That's your cold sores, that kind of thing. It can also affect other parts of your body, not just your lips, but it usually targets like really soft skin that's easy to penetrate. Um, uh, the varicella zoster virus, as you know, chicken pox, which also causes shingles. That one is also a type of polyhedral virus. Both of these are lysogenic viruses, which kind of also goes with that shape as well. And we'll talk about what lysogenic viruses are in just a moment. But this one, herpes, herpes simplex virus 1, herpes simplex virus 2, that one is genital herpes that is spread through sexual contact. And the varicella zoster virus, which causes chickenpox and shingles in adults, those are going to live in your nerve ending cells. And when you get really stressed out, that's when it goes through its lytic cycle. And we'll talk about that here in a minute. Abinovirus is a polyhedral capsid with a helical tail. This is what you think of. This is the shape you think of when you think of a virus. Like it kind of looks like the Mars, like or like a lunar landing type thing, right? You have your capsid here. You have like this rod shaped tail here, and then you have all these little spider looking legs. And your genetic information is going to be here. So this is an example. Our example is a bacteriophage. Um, they um, they are viruses that can infect bacteria. This one is for E. coli. This is bacteriophage T4. We actually use this a lot for molecular biology research because if you think about it, viruses are kind of amazing for genetic engineering and genetic research, right? Like we can manipulate these viruses to actually do things that we want it to do, right? Like inserting some genetic information that says, "Hey, E. coli, we don't want you to infect somebody and make them sick. We want you to pump out insulin so we don't have to get insulin from pigs anymore." And we can do that. And we have E. coli that actually grow and make insulin for us, and it's pretty amazing. So viruses aren't all bad, and we use them for research all the time. And I actually used some viruses and some of my lung cancer research as well. Very interesting stuff. Our other one is our helical virus. It's a helix, just like DNA. It's kind of like a double helix. It's like a little spring. And these are going to be mainly for plant families. This one's a tobacco mosaic virus. It infects members of certain tobacco type of the tobacco plant families. It can be very detrimental to farmers and stuff like that. So farmers aren't just worried about like pests, killing their crop. Viruses are also a major issue as well. So another way we can classify viruses is which host. And I kind of was talking about this. We have plant viruses that only infect plants, and then we have animal viruses that only infect certain types of animals. And we can even go further down and classify like this type of virus only infects dogs, or this type of virus only infects monkeys, or this type of virus only infects chickens. And so we can actually classify them based on which animal and which animal class they infect, and it can be even specific to a species. And then we also have, of course, bacterial viruses. It's important to know that these are called bacteriophages. Okay, so you need to make sure that you write that down. And I think I said that 3 times. So let's make sure we put a little star, highlight it, whatever, because that might most definitely be a question on a test. All right, so let's talk about our infection cycles. And looks like a lot to write down. And honestly, like write it down, think about it, but I'm going to show you pictures and that's going to make a lot more sense. So visually, it's going to make a lot more sense than me explaining to you with verbally. So the lytic cycle is the cycle that a virus enters the host cell, makes copies of itself, and then destroys the cell immediately. Okay. And a lysogenic cycle is a virus that integrates its DNA or its genetic information. It doesn't have to be a DNA. It could be any kind of genetic information, but we'll talk about that in a second. And it integrates its own genetic information into the host cell's DNA so that the virus's genetic information is copied along with the host cell, like silently. And then it's going to go through a lytic cycle when it's time to actually have like an outbreak or something like that. So remember I talked to you about how HSV1, which causes fever blisters, is a lysogenic virus. It stays mostly in lysogenic cycle, but when you have that fever blister, right, then it's going through its lytic cycle and it's actually it's replicating its virus and it's spreading, right? So they say that if you have a cold sore, you shouldn't kiss anybody or use the same like utensils and stuff like that because it's extremely contagious when you have a cold sore. and it's for life. And lysogenic viruses, you have them for the rest of your life, regardless if you're actually having an outbreak or showing symptoms at all. So it doesn't immediately destroy the cell. That's the thing about the lysogenic cycle. It's going to be kind of hidden. It's on the DL, right? Nobody knows it's there until it goes through its lytic cycle. So let's see what that looks like. So your lytic cycle is here, right? Your virus comes in, it attaches to the cell, it puts in its genetic information. It says, hey, cell, like you need to replicate my genetic information, not yours. And it has certain proteins that tell the cell to do that. Remember, proteins carry out all the functions of our cells. So whereas viruses don't have organelles, they do have proteins that tell our cell to make other proteins to help them replicate, right? So they start replicating all of our viral particles. We start putting them together, and then the cell, the host cell bursts, and more little. Each little virus is called a virion or virion, however you want to say it. Each little virion like goes everywhere and then goes to infect other cells around your first cell. Okay. And so remember, lytic cycle, virus comes in, starts replicating itself immediately is going to infect the cell and have some kind of symptom. So, and it's going to start replicating immediately. So this one currently that we're looking at is a bacteriophage, and that goes through its lytic cycle. And you can, if you want to write this down, that's fine. But honestly, just take a picture. You can do a screenshot, or you can not. And I'm gonna post this slideshow up on Google Classroom. All right, so lysogenic cycle. So let's think about our lysogenic viruses that we've talked about so far. So let's talk about HSV1 cold sores. Okay, so we have our cell, our host cell, we have our virus, it attaches to the host cell, then it enters our host cell. So notice the first three stages are almost exactly the same thing as the lytic cycle, but this is the fourth stage is where things start changing. Now we have what we call a prophage. A prophage is where the virus's genetic information is integrated with your genetic information. Okay. So we have prophage formation. So we see that the viral genetic information is being embedded into our genetic our DNA or the host cell DNA. Okay. Then we are going to just reproduce. So our host cells we're gonna go through mitosis, which is just a fancy science word for cell division. But we're secretly replicating, as you can see, we're kind of secretly replicating this viral genetic information, and our host cells do not know that this is happening. Okay. And then it's going to keep replicating and replicating. Now, if you're thinking, well, Miss Plizzy, why then how do we see symptoms if we never know it's there? Well, eventually a lysogenic virus does go through its lytic cycle, just like I'm going to show you right here. And so you can kind of combine these two cycles here. So once you keep reproducing your cells, there's something that's going to trigger that viral DNA to activate, or that viral genetic information to activate. So usually it's through some kind of stress response for HSV1, HSV2, or chickenpox, or shingles, because they live in your nerve ending cells. So a lot of times, like I know that I get cold sores, I get fever blisters. Everybody in my family gets fever blisters. So if my lips get sunburned, that's a kind of stress response because my lips are having to like repair, right? So I always use like SPF chapstick. If I'm super stressed out about school or work or what have you, or going through any kind of emotional distress, that's gonna cause a trigger. So for certain viruses, it might be different, but anything that involves like a nerve ending cell or something like that, most of the time it's going to be some kind of stress response that's going to trigger that lysogenic virus to go into its lytic cycle. So let's look at the two cycles together, okay? And I'm going to preface this with, remember, not all viruses are lysogenic, right? But all viruses go through a lytic cycle. Okay, so it's kind of like not all not all cacti or not all succulents are cacti, but all cacti are succulents, right? So we have not all viruses are lysogenic, right? But all um, viruses go through a lytic cycle at some point. Okay, so if you, let's say it's a bacteriophage and it's just going through its lytic cycle, right? So it's just going to go this way. It's going to embed it. It's going to put in its injected genetic information. Then it's going to immediately start replicating and making viral parts. Eventually, the cell lysis. Remember, lysis means to break. So that means like the host cell is gonna explode, and a bunch of little viruses or virions are gonna go everywhere, right, and infect more cells. Well, remember with lysogenic viruses, we're gonna actually integrate our viral DNA into our DNA. Remember, that's called a prophage. Okay, and we're gonna reproduce our host cells with that viral DNA or genetic information in it, and it's gonna reproduce normally. And then once it gets triggered, okay, it's going to say, okay, now I'm going through my lytic cycle, and all of those viruses are actually going to be able to spread. So let me use cold sores again. So remember, cold sores, you're gonna go like your virus comes in, injects its genetic information, okay. Then you have your genetic information from your virus and your own genetic information from your host cell. Those two get integrated to create what we call a prophage. Okay. Then your body is just going to replicate just like normal, or reproduce your cells just like normal. It has no idea that it's replicating all of that genetic information from the virus. And then there's going to be some kind of trigger. Okay. The trigger is like Miss Plizzy's trigger. Lips get sunburned, some kind of emotional distress, any kind of physical stress, mental stress can cause that trigger, right? And then it's going to go through its lytic cycle. And that is when I get the fever blister. That is when you see the symptoms. And that's also the most, that is when that virus is the most contagious, right? So it is you can spread HSV1 even when you do not have a cold sore or a fever blister. However, it is very rare and not likely. So there's that. Okay. So here are some examples of diseases that are caused by viruses. You do not have to know all of these. We're going to go through, I've kind of talked about chickenpox. I talked about HSV1, and we're going to talk a little bit about HIV and HIV/AIDS here in a second. But all of these diseases are caused by viruses. So if you want to take a look at those, it's quite interesting, and especially how they're transmitted. So before we start talking about specifically HIV/AIDS, let me tell you what a retrovirus is first, because HIV/AIDS is a retrovirus. And remember, retro means throwback, like we're going backwards, right? So this is why we call HIV/AIDS a retrovirus, because it contains RNA for its genetic information, but it's a lysogenic virus, right? So how is that virus going to embed its RNA, which is single-stranded, into our double-stranded DNA? Don't you think our body might be like, whoa, this part looks different. That's weird. Yes, absolutely it would, right? So in order for HIV to actually be a successful virus, it had to evolve a protein called reverse transcriptase. Now, reverse transcriptase, instead of going from DNA to RNA, reverse transcriptase goes from RNA and takes that RNA and makes double-stranded DNA. So it's going backwards. And we'll talk more about that when we talk about protein synthesis. Okay, so but going from RNA to DNA is backwards. And that's why we call it retro, because you throw it back, right? It's old school. So it's going to use that reverse transcriptase to actually turn its RNA into DNA and then embed its newly transcribed DNA into the host cell's DNA, which is kind of crazy when you think about it. Okay. And HIV is a virus that infects certain white blood cells, and it eventually causes AIDS, which we'll talk about the difference between those two in just a moment. Okay, so HIV and AIDS, also known as HIV/AIDS. All right, so first things first, HIV/AIDS, HIV specifically, AIDS is like a level of HIV, and we'll talk about that in a second. But HIV targets T cells, and T cells are part of what we call your adaptive immune system, that is utilized mostly for viruses. And the way they work is there's 3 different types. You have your helper T cells, which is your CD4 T cells. You have your cytotoxic T cells, which are also considered your CD8 T cells. And then you have your suppressor T cells. So your big one, though, you're like number 1 in that help detect infection of viruses and help make antibodies. And this is the one that we're really talking about when, especially in COVID, when you're like, oh, when you heard about T cell mediated response and all that stuff. Yes, this is the one we're talking about, helper T cells. When that helper T cell or a CD4 T cell comes in, it detects the infection, then it goes to the thymus. That's why they're called T cells, because thymus starts with T. And it says, hey, thymus, look what I found. This is bad. And then the thymus says, okay, we're going to make some B cells. B cells originate in the bone marrow, which is why they start with the letter B, as in Bravo, or bone. And your B cells are like, okay, I'm going to go find this. And then your B cell finds it, goes back to the thymus, says, hey, I found this. It is, in fact, an infection and bad. So now I'm going to become a plasma cell and secrete all of those antibodies. So obviously, super important, right? Cytotoxic T cells, they're like, hey, this is a virus infected cell. I'm going to attack it right now. It does it on its own. Suppressor T cells tell the immune system, hey, everything is chill now. Everything is good. We can calm down. Because if you don't tell your immune system to calm down, at some point you can go into this thing, what we call like a cytokine storm, which is basically a fancy science way of saying, hey, my immune system is overreacting and it will eventually cause death. And it's actually pretty quick, because your body just starts shutting down. Like all of your organs just start shutting down. So suppressor T cells are really important for like the end. Like, hey, the infection's over. We can go back to business as usual. Okay. So HIV stands for human immunodeficiency virus. The human, obviously, it infects humans. Immunodeficiency, it is attacking CD4 cells. So it is going to cause an immunodeficiency in that human, and it's a virus. Okay. So HIV, human immunodeficiency virus. And it attacks this helper T cells, the CD4 T cells of the immune system. And based on what I just told you about that, that's incredibly, incredibly bad, right? So I want to pose a question for you right now while you're thinking about it. Do you actually die from HIV or HIV/AIDS or that virus, or do you die from not having an immune system? I want you to think about that. We're going to come back to that question. AIDS. So common misconception is that HIV/AIDS are like different. No, it's a different, AIDS is a different stage of HIV. You can think of it as being like stage 4 of cancer, right? Like it's when it's like bad, like really bad. And so how we quantify this is that the person has lower than 200 cells per cubic millimeter, and is more susceptible to opportunistic infections because it only has like 200 CD4 T cells, okay, for each cubic millimeter, which is pretty much the same as like you have 200 cells per milliliter, right? Or I'm sorry, per microliter. Okay, so and a microliter is smaller than a milliliter. So remember um, your normal CD4 count is between 500 to 1,000. That's insane. That's what let's say it's 1,000, because that makes 200 quite easy, right? That is 1/5 of a normal person. Okay, so you have 1/5 of the CD4 T cells that you actually need. So this is really bad. So I posed you this question: do you die from the HIV virus, or do you die from another infection? So what I like to think of it, how I like to think of it is like, you don't like HIV is not going to kill you, but it will make it very difficult for you to just survive without being in a bubble, right? Because it's going to kill all of your CD4 T cells. So it causes what it causes is susceptibility to opportunistic infections. Okay, so that could be opportunistic infections is infections that are caused by a more opportunistic situation, right? People that are have a suppressed immune system, like people with HIV/AIDS, people that are on certain medications for autoimmune diseases, like me. You are more susceptible to opportunistic infections, and those can be caused by viruses, bacteria, fungi, um, even like ones that are normal in the body, part of our normal flora, like because we have tons of not tons of viruses, but we have tons of bacteria and we have tons of fungus that live in our body as normal, and it's a symbiotic relationship. A lot of bacteria help us digest food, and there are a lot of fungi that actually are a part of our natural flora as well. But people with weakened immune systems cannot balance that because they do not have such an important part of that, the CD4 T cell, that is such an important part of the immune system. So that's why they're called opportunistic, because they take advantage of that weakened immune system. So here's some examples. You don't necessarily need to know this, but they're very interesting. The cytomegalovirus, that causes pneumonia, which is in the lungs. Encephalitis is swelling in the brain. Gastroenteritis, obviously that has to do with gastro stuff, your tummy not feeling great. And retinitis, that is in your eyes. So this targets mostly people that have immunodeficiencies, obviously HIV/AIDS, and it also targets children, because that retinitis is going to be big with babies, because mothers can spread it to their babies. If you're a healthy individual, you will never know that you have this because you will never have any symptoms. You wouldn't know unless you gave birth to a child that had it, and then it was found out that way. Kaposi's sarcoma, cancer of the blood vessels, or any cancer for that matter, and this is just an example, any cancer, you're more likely to get have cancerous cells, or have cancer if you are immunosuppressed in some way. So that is a possibility. Pneumocystis pneumonia, also called PCP, that is caused by a fungus growing in the lungs. Not to be confused with the pneumonia, but it's a little bit different because it's a pneumonia caused by a fungus. Wasting syndrome, basically you lose more than 10% of your body weight, and lots of diarrhea, weakness, fever. That one's pretty bad. You just can't keep anything down. All right, so HIV transmission, these are important. Please write these down. It is the exchange of bodily fluids, specifically sexual body fluids or direct contact with blood. Okay, so you can transmit by unprotected sexual contact, pregnancy, childbirth, breastfeeding. Remember, it's going to cross that blood brain barrier, so any kind of anything to do with pregnancy, childbirth, or breastfeeding can transmit, and that's a big deal for mothers, new mothers with HIV. That can be very difficult not to spread that to the child or the fetus. Injection drug use, sharing needles, which is like a direct contact to like if you use a needle and then give somebody else, that's like direct blood contact. Blood transfusions and organ transplants. This is very rare now in the US because of screening, but back in the 80s, this happened a lot. So people that needed blood transfusions were getting infected, HIV infected blood transfusions, and it really increased the cases. Now we screen for that when people donate blood. All kinds of things, what blood type, all of that. But yeah, that's pretty rare these days, but it was a big deal back in the 80s. Occupational exposure, like if you're a healthcare worker and let's say you prick your finger with a syringe that you used on a patient that was HIV positive, you can definitely also get HIV. Now, just because you're exposed to it doesn't mean you're going to get it. For someone with a suppressed immune system already, like me, I'm way more susceptible if that were the case. Like if I would accidentally, prick my finger or something like that with a syringe that was used for somebody that was HIV positive. But just because you're exposed does not mean you will get it. Now, obviously, that increases your likelihood, but it's not 100%. Common myths. This is also important. The underlined ones are important. So being HIV positive is the same thing as having AIDS. No. HIV is that's the virus. That's what we call it. Okay. But remember, AIDS is a certain stage of that. So AIDS is remember, stage 4, like lung cancer. It is when it gets really, really bad. We say, okay, this person has AIDS to signify, hey, they're really susceptible to opportunistic infections. Okay. HIV can be transmitted by an insect or a mosquito. No, it cannot. HIV, you can get HIV from touching or kissing someone with HIV. No, you can't. Remember, it's the exchange of sexual body fluids or like direct blood contact. Okay. And I'm not at risk because I'm in a monogamous relationship. This is considered to be, HIV is considered to be an STI or STD, a sexually transmitted disease or a sexually transmitted infection. If you're in a monogamous relationship, you're less likely to get it as long as everybody is being in the relationship is being faithful. So just because you're in a monogamous relationship doesn't necessarily mean that you won't get it. Also, if you're in a monogamous relationship with someone who has HIV, you're way more likely to get it because it is sexually transmitted. Okay, so now let's talk about things that are pathogens, but are not viruses, but are also not cells. Okay, so a viroid, it's different than a virus because it has no capsid. It has to contain RNA. It will contain RNA, and it only causes plant diseases in angiosperms, or that's our fancy science word for flowering plants. So anything with a seed and fruit is an angiosperm. Apple tree, angiosperm. Okay, so viroids only cause diseases in plants that are angiosperms. We'll talk more about angiosperms and plants and stuff like that when we talk about taxonomy, ecology, plant science, botany. It's kind of fun. Prions Prions are like brain explode emoji crazy because they're nothing. They're just a protein. They're just a protein with the same amino acid sequence as one of our proteins in our body. I can't remember exactly which one for mad cow disease or chronic wasting disease. But it's just folded differently. Okay. And when our protein that's folded the correct way comes in contact with the prions folding way, our protein changes its conformation to be the prion way. Okay. This is going to cause encephalitis, brain swelling, brain hemorrhaging. It's happening mostly in the brain. Okay. For mad cow disease. And for most prions, when the way that you spread it is through ingesting neural matter. Yes, I said that. Ingesting like neural matter, so brains. Now you're like, Ms. Plizzy, how many people do you know that are eating brains? Well, let's talk about mad cow disease. So over in Europe, they do not have the same kind of regulations with beef, specifically ground beef and how it's made, as the United States does. And so in the early 90s, there was a situation where there was a lot of cow brains that were also getting mixed up with ground beef, and it was causing mad cow disease and a lot of humans that were not completely cooking. Remember, temperature denatures those proteins. You can denature a prion. It is just a protein, right? But these people were not cooking their meat all the way, right? I mean, hey, I like my steak medium rare. So, but these people were not cooking their burgers and their ground beef all the way, and they were getting mad cow disease that was causing brain hemorrhaging. It caught, and then like the brain hemorrhaging and the brain swelling. It also causes like you to like waste away. And chronic wasting disease in deer is basically the same thing as mad cow disease in humans, but for deer. And we see that all the time here in Texas, in the deer populations here. Once it happens, once one deer like gets sick, usually quite a few do, but it's in little pockets. So definitely interesting. So let's keep going. So just some statistics. An estimated 44,000 Americans are newly infected with HIV each year. You can prevent that by having protected sex, not sharing, not having blood to blood contact. If you get a blood transfusion, you really can't help that. But the United States is really, really good about regulating and testing for HIV in their blood donations. So that's not really anything to worry about. There are approximately 1.2 million Americans who are living with HIV/AIDS. People are living with HIV/AIDS longer and longer because we have so many different medications that do not cure it, but deal with the symptoms and help create a better immune system and help protect some of those CD4 T cells that they have left. But it's very, very difficult to come back once you get into AIDS. It's very difficult to get back into just HIV after you've gotten that low count of T cells. The CDC estimates that 13% of HIV positive don't know they are infected, meaning they may be transmitting HIV without knowing it. And that's the super important part. Like, get tested, know, and have, since this is a sexually transmitted disease, these people need to make sure they're tested, that they know, and if they do know that they are having protected sex, and to also make sure that they are having protected sex no matter what, even if they don't know, because then that also decreases your chances. Okay, so that's viruses. Very interesting on topics. So that's all I got. I will talk at you later. Bye.